Okay, good morning, good evening, good afternoon, wherever you are in the world, to everybody who's attending here. Thank you very much for coming to this Validate seminar. And my name is Blakely Nixon. I'm the Communications and Events Coordinator at Validate. And I'm going to introduce our amazing speaker for this event. So Professor Annemieke, and here we go, Annemieke Geluk is the head of immunotherapy department at Leiden University Leiden University Medical Center in the Netherlands. Her work spans basic, translational, applied, and field research, and her research combines lab and field work, and she's also a huge proponent of the One Health and the global health approaches to research, which is an approach to health that acknowledges the interconnectivity of humans, the environment, and animals. And today, Annemieke is going to be talking about use cases for immunodiagnostic tests for leprosy. So I'm going to hand over to our amazing speaker, and thank you very much for coming. Thanks, Anne.
Thank you, Blakely, and also thank you for the invitation to share some of the work of my group on research on leprosy today. And as some of you may know, so I work at the Leiden University Medical Center, and within the research of the mycobacterial disease group that I'm in, we focus on vaccines, pathogens, drugs, and diagnostics for leprosy and tuberculosis. And if you want to find more, please check this link here below. And today I'll only focus on the diagnostics for leprosy. And as you may know also from tuberculosis research, there is a diagnostic need for the early detection of tuberculosis as well as leprosy cases. Because, for instance, for tuberculosis, and these are data from 2023 from WHO, it's known that there are about 11 million patients per year, but that around 2.7 million of those are undiagnosed. So they go out and they transmit and they perpetuate the disease in this case. And for leprosy, it's known that for years already, the annual number of new cases stagnates around 200,000 with a little dip during the COVID pandemic. And also that 1 in 10 of the newly diagnosed patients are children. So this also indicates that transmission is ongoing. And therefore, what is the diagnostic need for both of these diseases is a rapid diagnostic test that is field friendly, globally applicable, and preferably in low resource settings. And so therefore, WHO, during their UN high-level meeting on TB, they claimed that there should be a rapid test for TB by the year 2027. And also, WHO is working on TPPs for leprosy diagnostics, also including diagnostic tests for early diagnosis of the disease that should be rapid, field-friendly, and globally applicable in low research settings. Because as you can see here in the areas where these diseases are endemic, they are usually in the global south and usually also in niches in those countries where there is little or no resources to have, for instance, like laboratory settings. So during this presentation, I will focus on leprosy and not on TB. And if you're interested in knowing more about leprosy, last year I published together with a group of experts in different fields of leprosy in Prime Views Leprosy for Nature Reviews Disease Primers. You can look that up for more information on leprosy and multiple aspects of it. And first of all, for those who are not that familiar with leprosy and work preferably on TB, leprosy is a poverty-associated neglected tropical disease. So for instance, funding, this means that we do not have access to some of the funding that is available for instance, TB or for malaria or HIV. But it does rank second after TB as a severe human mycobacterial disease. It can be caused by Mycobacterium leprae, but also by Mycobacterium lepromatosis, which is, but it's much less contagious than a Mycobacterium tuberculosis. It affects not the lungs, but the skin and the nerves, and usually this results in irreversible handicaps that a lot of people are familiar with, that are depicted here on the right, for instance. Well, the mycobacterium that causes the leprosy, so Mycobacterium leprae in most cases, is much less virulent than MTB, and it grows at different temperatures, optimally at 35 degrees, so lower temperatures. Hence, it occurs more in the extremities in humans. It has the longest double time of any mycobacterium, 14 days, but because it lost a lot of genes that are absolutely required for survival, it requires a host to survive. So it does not grow in vitro, and many people have tried that. But nowadays, for instance, armadillos or nude mice are used to produce live Mycobacterium leprae. So this impedes a lot of the research and also research on diagnostics. What a lot of people do not know or do not realize actually that we have learned a lot from leprosy, and therefore I always like to make this statement: leprosy first, and it's not like a political tinted statement, but it is a scientific statement because approximately 10 years before Robert Koch detected Mycobacterium TB, Armauer Hansen already detected Mycobacterium leprae as a pathogen for human disease, and also in other aspects. For instance, the association of HLA and disease was identified first using leprosy patients, and it was by Jon van Rood in the 70s. And also the regulatory T cells in humans, Tregs for short, they were identified in the 80s, also using leprosy patients, as leprosy is kind of model disease. And finally, also for the Th1, Th2 dualism in immunity against Mycobacterium leprae, that was also first discovered for humans in leprosy patients. So we could and we, I mean, then the mycobacterial research community focusing mostly on tuberculosis. So we could use Mycobacterium leprae and leprosy disease as a model disease for various instances. And there's something else that is unique about leprosy, and that is that it's a spectral disease. So on one hand, there is the paucibacillary granulomatous disease, with a lot of inflammatory cytokines and causing a kind of autoimmune disease, and with a lot of cellular immunity directed also against the host. And on the other hand, there's multibacillary disseminated disease, and with a lot of bacteria, a lot of antibodies, which are not protective, by the way. And that causes a lot of nerve damage in another way. Actually, the multibacillary disseminated disease is usually referred to as the more severe type of leprosy, but also paucibacillary leprosy can cause nerve damage, leading to irreversible handicaps. In between, there is a lot going on actually, because from my field work, we rarely actually detect pure paucibacillary or pure multibacillary, so like the lepromatous part of the spectrum or the CT part of the spectrum, but usually it's somewhere in between. So these individuals in between have a little bit of humoral immunity and a little bit of cellular immunity, or a mix of that. And this is important because it also affects the way that we can use diagnostics for detection of disease. And I just showed you that there is here I just showed you that there is a division in paucibacillary and multibacillary. So actually, Th1 goes with paucibacillary, or Th2 cell response with multibacillary leprosy, but it's not such a black and white division. And also what we also published last year in this publication that's depicted in this slide, it goes beyond Th1, Th2 duals. And there are many more T cell subsets that are in that are involved causing pathology or protection. And you can read up on this a little bit more in this publication. And so currently, leprosy diagnostics, a diagnosis of leprosy is pure clinical, meaning clinicians in the field, they look at the clinical signs, and there are no really good diagnostic tests that can detect leprosy and either palsy or multibacillary leprosy. The test that is used, for instance, in Brazil a little bit, is a visual lateral flow test that detects antibodies against Mycobacterium leprae. And that is used actually more as an adjunct diagnostic test. And I'll get back to that a little bit later. So what we would like to have is a test for early diagnosis so that we can prevent disease and perhaps also reduce transmission, but also a test to classify who is MB and who is PB leprosy, because PB leprosy patients are treated less long. So for instance, about 6 months, whereas multibacillary patients, they are treated for about a year. And so classification is important in the field, and also detection of Mycobacterium leprae infections. So if we have, because it takes a long time for Mycobacterium leprae infection to really cause the disease that is clinical detectable, we would like to also early diagnose infection so that we can prevent this infection proceeding to disease. And as a fourth use case, there is monitoring of treatment. Because, for instance, if a person is diagnosed with leprosy, but not at early stages, then treatment can become very complicated, and leprosy reactions can also occur. And this will then, if we can monitor this treatment and monitor also detection of development of leprosy reactions using a diagnostic test as an adjunct diagnostic tool, in addition to clinical diagnosis and clinical monitoring, this would help to see whether a patient is responding to a certain treatment. And especially in these difficult to treat cases in which leprosy reactions occur, this is a diagnostic need. And finally, what has been approved and endorsed by WHO since 2012 is so-called PEP. And PEP means prevention of leprosy by providing a prophylactic dose of antibiotics. And usually this is given in a single dose, and for instance, a post-exposure prophylaxis given to contacts of leprosy without any signs and symptoms of the disease yet is an example of that is a single dose rifampin, which has been endorsed by WHO since 2012. And this is given to individuals to prevent leprosy and overall also to reduce transmission. But although we know that in some of the studies that have been studying PEP with a single dose of rifampin, that the number of new cases resulting in the area where PEP was given decreased, we do not know actually whether the transmission in an area where PEP is provided is also decreased. And we also do not know whether when a single dose rifampin is given, whether that is sufficient and what it does to a person's immune system or immune status at an individual level. So this is not known, despite the fact that we do know that PEP can reduce the number of leprosy new cases. But because there is this prolonged period in which leprosy infection can develop until disease, we would like to also monitor the responses to PEP directly, and I will show you some examples of that later on. So these are the use cases that nowadays are considered to be required in leprosy healthcare. And as I already showed, we're working on target product profiles for leprosy diagnostics for these several use cases. And at the LUMC, we have chosen to work on immunodiagnostic tests for mycobacterial diseases, and in particular for leprosy. And why is that? Now, that is because, as I just discussed, leprosy has this unique disease outcome, in which is either multibacillary or paucibacillary disease outcome, and this immunity parallels the outcome after infection. So when the host is invaded by mycobacteria, a lot of cells are involved in the immune response, and depending on the genetics, the health status, etc., of this individual, the immune response is produced and is also measurable as a kind of footprint of the infection or previous infection or disease, even when the pathogen has been cleared, so when the person is PCR negative. And this results in the production in the blood, or sometimes also in urine, of immune biomarkers that we can measure and that we can compare between individuals, so between infected and clinical disease, and also intra-individual. So having whether a person is progressing to disease or whether a person is efficiently treated. So intra-individual, these biomarkers can differ as well as inter-individual. And that is what we have been working on for a long time. And if you know a little bit about biomarkers and the biomarker pipeline, so usually you start with many candidates during the exploratory discovery stage. And a few samples only that are derived from clinically well-diagnosed patients. And then some of the candidates that look promising are confirmed in a little bit more samples. Then validation should take place with less candidates, but even more samples, and preferably at a global level, so including not only one area where leprosy is endemic, but multiple areas, and also longitudinally, so including either contacts that are followed up in time, or including patients that are being treated. And then, of course, with a subset of these candidates, so less than 10, we have been doing field trials with thousands of samples and prospective follow-up. And finally, the biomarkers of choice are being applied to a diagnostic platform to be used as diagnostic test or predictive test or even therapeutic test. And most of the work that we have been doing as the LMC team on host biomarker identification for leprosy was done in Bangladesh. And as you can see here in the picture, there are many titles of the projects that have been going on since 2012. And also currently there's a study ongoing now that includes either BCG vaccination or as single dose rifampin, or only sampling of blood and follow-up of these contacts and leprosy patients. And using about 7,000 individuals in these studies, we have been identifying a lot of biomarkers. So biomarkers for infection, biomarkers for disease, biomarkers for prediction of paucibacillary leprosy even. And of course, you don't only want these biomarkers to be used for one point, one area in one country. So we have also been working in other areas like Brazil and Bolivia, Indonesia, India, Madagascar, also in China and Ethiopia in the past, and also in non or much less endemic areas where leprosy also occurred, such as the Netherlands and the USA. And the overall way in which we work was, of course, to have the patients, leprosy patients, centrally. So starting point and end point are patients. And this was done by active case finding approach by the Rural Health Program in Nilphamari in Northwest Bangladesh. And this went along with field sampling and biobanking of samples. Because for leprosy, it is always difficult to predict who is going to develop leprosy, because a lot of people, a lot of the contacts will never develop leprosy. So we have sampled a lot of these contacts, whereas only a few cases in those 10 years that we've been working there do appear. So it is a matter of patience, actually, but it goes hand in hand also with capacity sharing. So training of the staff, training them sometimes in PhD sandwich programs, and developing the test as we go. So developing from laboratory-based until more field-based tests. And of course, we did a lot of omics, so there was transcriptomics or proteomics and metabolomics ongoing, and which led to the identification of multiple biomarkers that we have used for implementation in diagnostic tests. And I will get to that a little bit later. So most of the work has been done on proteomics, and that was because 10 years ago it was more feasible to apply identified proteins as biomarkers in low complexity diagnostic tests. Nowadays, actually, and we published this recently in a large consortium on TB, it's also it's also possible to use a finger prick sample of blood for gene expression analysis, so transcriptomic biomarkers. And so actually, we're also trying to find out whether that would work for leprosy, but I will not focus on that in this presentation. So we did most of the multiplex cytokine analysis either by Olink or by Luminex, and identified several biomarkers. But we're also currently working on more extensive immunophenotyping, especially for the paucibacillary cases, as well as the bacterial killing capacity. I'm also not going to talk about that, but hopefully in future a colleague of mine, Anouk van Hooy, will be able to do that here on the Validate seminar. Actually, so there she is. Anouk worked as a PhD student in my group, and she did a lot of these multiple host biomarker analysis. And what we found is that some of the biomarkers are shown here can be useful for both multibacillary patients as well as paucibacillary patients. Some of them only for the paucibacillary patients, and some of them only for multibacillary patients. So if you combine these biomarkers, then you can have an increased diagnostic value for both identifying both multibacillary and paucibacillary leprosy. What I also need to say is, and what is depicted below here, in analogy to the quantiferon TB test, in which there's an overnight incubation period to stimulate the immune cells in blood with specific leprae antigens, we were also able to identify biomarkers that are not measured directly in serum or fingerstick blood, but if you incubate blood overnight with these leprae unique antigens, then some of these markers can be used also to identify paucibacillary disease. So conclusive, it's important for especially paucibacillary disease to detect it, to combine multiple biomarkers, and if necessary, also to have an overnight incubation step included, just like in the quantiferon TB test. So having identified biomarkers that would work for paucibacillary and multibacillary disease is one thing, but of course we want to go from the lab to the field. And the way we do it, and you may be familiar with that already because we have been working on that for a while, is so-called UCP lateral flow assay. And UCP stands for upconverting reporter particles, and these are rare earth particles that are not available in the environment, and therefore they are very sensitive because they have a low background, and they allow us to have a quantitative measurement as well. And furthermore, they're very stable, so the tests do not need a cold chain during transport or during field work. And the only thing we do need is a portable reader. And one example of that is depicted here on the right. And the way this works, this upconversion process works, is as follows. The upconverting molecules, which are attached to an antibody or linked to an antibody of choice, of whatever biomarker you want to detect. They are activated by a infrared beam, which is in the reader. And upon activation, the structure of the upconverting particles emits in the visible spectrum, which is again detected by the reader. So the reader provides the signal and also detects the emitted signal, and thereby quantifies the number of molecules of your biomarker of interest. This therefore combines actually both the characteristics of an ELISA and the characteristics of the so-called ML flow or lateral flow test. So it's the best of both worlds, because the ELISA is quantitative, whereas the lateral flow usually is only qualitative. But the ELISA is lab-based, whereas lateral flow can also be used with capillary blood and is field-friendly. And the ELISA is operator independent and gives a quantitative measurement, whereas the lateral flow cannot. But the UCP lateral flow assay combines this, so it remains quantitative while being field-friendly and can be used with fingerstick blood, and gives an operator independent outcome. So we have so far developed this for single and for multi-biomarker tests. And the most advanced test is the one that measures one single biomarker, which is depicted here on the left, and using a lancet and a finger and a so-called minivet, which with capillary effect can sample just 20 microliters of fingerstick blood exactly, so it's a measured amount, and some of the buffer in this little flask. We can perform this assay in the field and use the reader also, the portable reader also in the field, to detect a quantitative a quantitative amount of the biomarker of interest. And what is depicted here is a test for an antibody directed against PD-L1 of Mycobacterium leprae. And for a multi-biomarker test, we are working on this kind of platform, which you can, for instance, measure also with fingerstick blood and with buffer in a minivet. You can measure 4 biomarkers at the same time. The reader will be a little bit different, but this is an ongoing process for leprosy. Well, as I already indicated, so the most advanced biomarker is the anti-PD-L1 antibody. And we have chosen for this test because this antibody, there's a strong correlation between the amount of antibody and the amount of bacteria that is infecting an individual. And this has been shown by many other groups a long time ago. And whether there is an infected individual or an individual that is carrying Mycobacterium leprae in slit skin in the ears. Both indicate a good correlation between anti-PD-L1 and the bacterial load. And also the number of these antibodies is reduced in multibacillary patients upon efficient treatment. And this has also been shown by various individuals, various research groups. And it works a little bit like this. So I already indicated there is a disease spectrum in leprosy. So if an individual is leprae exposed, most of them stay seronegative because innate immunity directly kills mycobacteria. Whereas some of the individuals, they can become infected but they can still, if the bacterial load is low, can still remain anti-PD-L1 seronegative because they have only a low number of bacteria. However, if the bacteria starts multiplying in the individual becomes leprae infected and anti-PD-L1 seropositive. But the number of individuals that do carry a lot of bacteria is much less than the number of exposed individuals because most people can just cope with leprae by their innate immune system. Recently, so last year, we published a paper on our diagnostic test and also indicating that it's now ready for use as a practical tool for leprosy for several use cases. We have a version that we used first for several studies in the field, which is depicted on the left, but the version 2 now is also available and has a biodegradable format because this, in collaboration with a startup company called OCOS, we are now producing this type of test, which is a plant-based cassette. Anyhow, I promised to say something about the use cases of our immunodiagnostic test for leprosy. And as I told you that we have this test that detects antibodies against Mycobacterium leprae phenolic glycolipid 1, so PD-L1. And this test detects both present infection as well as past infection because these antibodies, and we do not know exactly what's the mechanism behind it, but these antibodies, they can remain in an individual, an untreated individual, for a long time. So the presence of these antibodies indicates the person has been infected or is infected, but not when. So cannot say anything about how long ago this has taken place. So if you, however, measure antibodies in children, then we know that this has happened a relatively recent time ago because it cannot exceed their age. So what we have been doing in a door-to-door survey in Bihar, India, is a field-friendly sero-survey in these children to monitor M-leprae anti-PGL1 seropositivity, and use that as an indicator for transmission of Mycobacterium leprae in that area. And what we saw is that in healthy young children, so their age ranged about 5 until 10 years, there was about 12% sero prevalence in these areas. As in addition, what we also detected is because we can measure this quantitatively, some of the children which had a high level of antibodies but no clinical signs yet were revisited the year after, and this took place in 2020 and 2021. So you can imagine during the COVID pandemic that it was a bit difficult, but we went back to visit the children with the high levels, and some of them were then also diagnosed with paucibacillary leprosy. So although this was not the main research question of our study, it is possible to also early detect leprosy, even paucibacillary leprosy, using this diagnostic test for antibodies against PGL1. In addition to that, you may have heard that also, for instance, by the exposure to infected armadillos, because armadillos are are animals that can carry Mycobacterium leprae and that occur in the southern states of the United States. And in 2010, it was already published by Dick Truman and others that transmission of these animals occurred to individuals that lived in the southern states of the United States. And therefore, it will also be of interest to use this test in individuals without any clinical sign, just to see whether there's also transmission in a non-endemic population. Another use case of this test is a One Health approach. And for that, we have been working together with the University of Edinburgh, actually Roslin Institute, and because they identified together with Charlotte Avanci in 2016 already that the red squirrels are infected with Mycobacterium leprae as well as Mycobacterium lepromatosis. And we wanted to see whether we could detect in these animals also quantitative levels of these antibodies. And we have described this as well. And recently, we have done a little bit more of that research. Unfortunately, it's a protected species, so we cannot do a lot of research on these animals and usually are dependent on roadkills. But we did see that also after 2016 and after that publication that still there are animals that are antibody positive. So it's still being transmitted in these animals. So therefore it would be a useful tool also to do some surveys with amongst these animals. Same goes for the detection of Mycobacterium leprae in the 9-banded armadillo. And that's a study we did together with the National Hansen Disease Program in Louisiana, in Baton Rouge. And together with a group of Linda Adams and Ramanuj Lahiri, who have a experimental model and in which they test Mycobacterium leprae infection in armadillos because they're susceptible animals and even show a similar spectrum as humans do. And we also could use the antibody test. And what we showed in this study is that the number of antibodies is rapidly increased in animals that are susceptible, whether the resistant animals that do not have any bacteria and do not show any disease even after a year, they do not show any antibodies either. So if there is a program in which armadillos in the wild are being surveyed for the presence of Mycobacterium leprae, because they cause transmission in the area, may cause transmission in the area, then also detecting the levels in these animals with our diagnostic test will be feasible, even cross a species barrier. And so in summary, it works as follows in humans, using a 20 microliter finger thick blood sample, a little bit of assay buffer, and a lateral flow cassette. We have to wait around 10 to 20 minutes and can directly measure quantitative levels of the antibody. And we can also use, if you set a threshold, so you can use then as a qualitative measurement. But the result is quantitative measurements. And then we can use it for diagnosis, for measuring transmission, for monitoring interventions such as PEP, but also monitoring treatment in patients, and to measure infection in animals. Well, as I just told you as well, is that we have to deal with the leprosy disease spectrum for diagnosis of leprosy, and we identified several markers. So we also need a test that can measure at the same time, in the same sample, multiple markers. And for that, we are currently, for instance, working on multiple biomarkers, detection of multiple biomarkers for detecting the effect of, or monitoring the effect of post-exposure prophylaxis study in Nilphamari in Bangladesh, because there is no direct evidence available, despite that SDR-PEP is WHO endorsed. There's no direct evidence for the effect of a single dose of rifampin on leprae infection, either in the short or in the long run. And so it will be useful to measure, for instance, with a finger a stick sample, who needs extra PEP or who does not need PEP. So therefore, we have been, since 2019 already, immunomonitoring the effect of a single dose rifampin, a single double dose rifampin, and a little bit recent, more recent, on PEP plus regimen, which has, besides rifampin, also clofazimine. And in another study, we will also focus on the effect of bedequalin as post-exposure prophylaxis, using several markers. And the way we go about this in Bangladesh is as follows. So there is an active case finding program, and when an active case, a multibacillary case, is detected, the patient is of course treated using routine MDT, but the contact of the index case are given randomized, are given SDR, SDDR, PEP++. And we measure the biomarkers. We take finger sticks blood and measure about 6 biomarkers at T0, 2 weeks after PEP, 4 weeks after PEP, 8 weeks after PEP, and after 6 months. And after a year, when the multibacillary case is assessed, and also there is a clinical assessment of the contact cases. And we do this is all done by the field team themselves. They take finger stick samples, and they are testing the UCP lateral flow tests. So finally, to give you a summary, we have been working so far and are still working on the biomarkers and immunodiagnostic tests for diagnosis of paucibacillary and multibacillary leprosy. We're also in various areas monitoring transmission using antibody seroprevalence of anti-PGL1 antibodies in young children, young healthy children, but are also looking at the intervention effect using multiple biomarkers and changes over time, either in individuals or at a population level. And therefore, I would like to thank some of our collaborators, particularly people in Bangladesh, but also a lot of collaborators at the Erasmus MC in Rotterdam, because we did a lot of the studies together with them since 2012 in Bangladesh. Also people from the Marcelo Mira group in Brazil, Diana Hege in Nepal, the National Hansen Disease Program group in Louisiana, and the group in Edinburgh. And finally, also the team of the UCP lateral flow assay development at the LUMC, and of course, all of the funders that made this work feasible. Thank you very much for your attention. I'd be happy to have any questions.
Can I ask, Annemieke, can you stop sharing the slides? Because I think it's nice to be able to see people. So does anybody have any questions? Please turn on your camera. Bar and Paul, are you lining up for a question there? And right, we're going to start with T. T put her hands up first. So if you want to ask a question, do what T did and raise your hands. Oh, no, it was a, yeah. Was that, oh, sorry, it wasn't T. T was applauding. So congratulations. Tahina, would you like to ask a question? Tahina, can you hear us? And Gabriel then.
He's muted.
Oh, there we go.
Can you hear me now?
Yes.
Perfect. Wonderful presentation. I just have one question. You said you collect samples from these leprosy patients to identify whether they are MB or PBs. What samples do you collect, apart from the blood, obviously?
Okay, no. So to avoid any confusion during the biomarker identification phase, we use blood, so venous blood and finger stick blood of cases that have been clinically diagnosed. So that is, so we are not using the test for diagnosis yet. The test is being developed using the clinic as a starting point. So that's one thing. So we use mostly venous blood and later on finger stick blood, because we aim for a test that is to be used in the field and that can be used by anybody with only a little bit of training.
Perfect. I have one more question, if that's okay.
If it's a quick one.
Oh, okay. So there was a slide where you also said there was bacterial killing capacity was measured. So is that a simple midget that you do?
It's not simple, but
for leprae, obviously not simple.
Sorry.
For M. leprae, obviously it's not that simple.
No, but it's also not for Mycobacterium leprae. It's not possible because a midget would not, it wouldn't grow. So we are working on a assay, and actually that's Anouk van Hooy who can do this, because she has recently received a Validate pump priming grant, and so that is also why I touched upon it, but I will not go into detail because she will be able to do that hopefully sometime next year to discuss about that. But it's a midget, yeah.
Oh, thank you.
Okay, you're welcome.
Thank you for plugging the Validate Pump Priming grants, which are available to Validate members. So if you're not a member, you should join and be able to apply. Gabriel, I think you're next.
Hello, can you hear me or see me?
Yes, we can, yes.
Perfect. Thanks very much, Annemiek. That was really a really great talk, a great summary. I was curious about your biomarkers, the anti-PGL1 biomarkers you described. I wonder, because you show data on how it relates to bacillary burden and reduces on treatment, I wonder whether you'd explored at all its relationship to the risk of developing reactions in these in patients, and also the sort of subsidiary to that. What happens to levels when someone develops a reaction? Because perhaps bacillary burden doesn't go up, but there's a state of sort of hyperinflammation. I wonder whether they relate in any way to severity or extent of reactions. Thank you.
Thank you, Gabriel, for that question. So we and others have shown that, I mean, the more bacteria a person carries, the greater the risk upon treatment that the individual develops a reaction. And of course, the more the bacteria they carry, the higher the anti-PGL1 levels are. So in that sense, I mean, that's the correlation. So in that sense, you can use it as a risk marker for individuals developing a reaction. And the other question, does the level of antibody change during the reaction? We were able to follow up a case, a leprosy case in the Netherlands, who developed a leprosy reaction, and so in detail, and we published this a few years ago, and we saw that PGL1 did not, it was not, the reaction occurring did not affect PGL1. And other biomarkers were affected, of course, inflammatory markers, etc. And we could follow treatment very, very well with that. PGL1 is a risk one, but it does not, well, you cannot detect reactions with it.
Lovely. Thank you very much. Thank you very much.
Okay.
Hi, and Lydia, do you have a question?
Yes, I do.
Go ahead.
Wonderful talk. About the biomarker discovery, I know you had mentioned that you used blood in that stage. What are your thoughts about using actual lesion samples, if possible, like samples directly from the skin, where you would have sort of expression of the leprosy symptoms? Please, did you hear the question?
Yeah, I heard your question. Thank you for that. That's interesting. Of course, we are focused at designing a test in such a way that it is very easy and to perform. I think also some of these use cases are for infection, and then the individuals that you are screening, they don't have any signs. So what you are suggesting would only make sense if there are individuals who have patches, so for diagnosis. And what is usually done by leprosy clinicians is that they take a small biopsy or sometimes even a scraping and do a qPCR and to verify whether this is M. leprae or M. lepromatosis or another skin disease. And that is usually done in a molecular biological way. So the presence of the markers that we are looking at, I mean, they would not be present on the skin. Does it answer your question?
Yes, it does. Yes, it does. Thank you very much.
Okay.
Thank you, Lydia. Before I come to Hugh, we've got a question from Mohammed in the chat. This is Mohammed Uddin from ICDDR. I think you worked with him in 2012, he says. And he compliments the presentation, but he says he's really interested in the transmission of leprosy among contexts. How was it diagnosed, and how did you handle treatment monitoring?
So transmission of Mycobacterium leprae in contacts. I mean, what we are doing now, and this is recently set up also in Bangladesh, is detecting antibodies in young children. And that is how we measure transmission in the population as a measure. So a seroprevalence in the population in young children is a measure for transmission in the area. I'm not sure what Mohammed means with transmission in contacts. Maybe.
What he actually said was, can you speak up, Mohammed? I'm really interested in transmission of leprosy among contexts, how it was diagnosed, and treatment monitoring. Question mark. That's how he put it. So, there he is.
Thank you very much for the nice presentation, and I'm really glad to see you after a long time. And I also heard about the contact tracing among the leprosy patient in Bangladesh from the institute. They're doing these things. So I just want to know about the transmission from leprosy patient to contacts if they're positive in antibodies. Do you have any tools for identifying the contact tracing the patients? The leprosy is really transmitted from the patient, like mother or father to the child. These things.
So actually, this is part of what we have been doing in the active case finding studies. All of the studies in Nilphamari were active case finding based, and then focused on the context of the new cases. And so we are still working on that. And so post-exposure prophylaxis is given to the contacts, and we are identifying the new cases clinically. So in order to identify new good biomarkers, we are dependent also on clinical diagnosis. I'm not sure whether that answers your question. Anyhow, so we are now also conducting a serosurvey in the population in that in Nilphamari amongst children to see whether post-exposure prophylaxis, either given as a blanket or given on contacts only, is reducing the number of children that is sero positive. Does that answer your question?
Yeah, I got the answer. Yeah, thank you very much.
Okay, thank you.
Anamika, we've obviously reached 11 o'clock. Are you happy to answer a couple more questions, or do you need to go?
Sure, no problem.
Okay, we'll press on. If anybody needs to leave, you won't be offending anybody, because it is 11 o'clock. It's at the top of the hour UK time. So I think we should go with Hua first, if you have a question, and then there's a couple in the chat as well.
Yes, thank you so much for the wonderful talk. I really enjoyed it. There is quite exciting data that you have, especially with different populations and a location. And I'm wondering if there's any correlation that you saw with the leprosy disease spectrum in regards to if you also take patient genetics in consideration as well.
Yeah, that's a nice question. So I'm not a geneticist, but we work together with Marcelo and Mira also, who obviously has a lot of experience there, and working with Erwin Schur also. And the cases that we study are, I mean, we're dependent on the way that cases are detected in the areas, so the institute that we collaborate with. So for Nilphamari, for instance, this has been mostly paucibacillary cases because there's an active case finding program. I'm not sure whether you can directly compare the genetics then of those cases with the cases that we find in other areas where there is no active case finding and they are self-reporting, because then they're usually multibacillary cases. So you may know from the literature, and otherwise you can look this up in these publications, that there are genetic associations, but we haven't studied that in real great detail, other than a recent publication that we did in collaboration with Marcelo Mira also, in which we showed that not a genome-wide screening, but targeted analysis of some of the genes. There were susceptibility genes for leprosy in Brazil, also turned out to be susceptibility genes in the families that we had in Nilphamari, in Bangladesh. So different population, completely different, but the same genes that were important.
Amazing. Thank you so much.
Okay.
We have one question in the chat from Kidist. I'm going to give you 10 seconds to come on camera to ask it in person, Kidist. But if not, I will ask it the best I can. He says, my question is regarding the use of finger prick, which is important in having a friendly assay, a field-friendly assay in relation to specific, not anti-PGL, but other biomarkers. How do you manage that? If you check in the chat, it perhaps is a bit more coherent. If you
well, it would be nice to see Kidist. She did a PhD in my group, so I hope that she could come on camera. Kidist, can you? Ah, nice to see you. So you were asking about the other biomarkers, how we detect them.
Yeah, I'm more concerned about the specificity, because anti-PGL1 is fine with the finger prick, but for the other biomarkers, the specificity might be an issue. And how did you manage that?
Yeah.
And glad to see you, by the way, and glad to see all the.
Yeah, nice to see you. Yeah. So anyhow, this is a good question. I'm glad you asked this. Yes, some of these markers definitely are not specific for leprosy. So therefore, it's some of the points are very important. First of all, we sometimes use the incubation step with leprae unique antigens, which provides specifically in paucibacillary cases more of the cytokines. And because we work with a quantitative test, you can have like a threshold above which this is specific for the disease. Furthermore, so besides having quantitative measurements, besides having also PGL1 as an add-on, and as well as the cutoff availability, and we combine the biomarker profiles, because what we see, for instance, also in our TB work, is that combination of these biomarkers and putting them on a certain threshold above or below which they are specific, that helps a lot to identify in particularly paucibacillary cases from the contacts, because these two, the paucibacillary cases and contacts who are heavily exposed for sometimes many years, they are most similar, and those are the ones that we want to discriminate. And for that, the combination of biomarkers, the levels of biomarkers is doing the trick, so to speak. But I also want to say that the current biomarkers that we have done using exploratory platforms that were available, such as Luminix and Olink, they mostly have identified inflammatory markers. And now we're still working with a new PhD student, also working on the detection of other biomarkers for especially paucibacillary cases, as well as, for instance, pure neural cases. And so I think the strength is quantitative measurement of multiple biomarkers that is helping us to do this.
Okay, I think that's a really good place to end. We have gone over a little bit. So my favorite thing about doing leprosy talks is it's such a neglected pathogen that everybody knows everyone else who studies leprosy, because it's not a giant community. You don't get that so much with TB, where there's quite a lot of researchers, but it just shows you how it is one of the more neglected of our neglected pathogens that we study here at Validate. So what I want to do now is just thank Dr. Cheluk, Professor Dr. Cheluk, and if everybody could show their appreciation by either just sort of waving at the camera or using the react button like this, for example. There we go. Like Hawaz's done, yes. And just to say thank you for an excellent talk. And thank you for some excellent questions as well. I'm glad we were able to go over a little bit so everybody gets it. So having said that, so I'm going to wrap this up. I'm going to stop the recording, but thank you very much, everyone, for coming.